Okay, so I am going to check a flow cell. In this video, I'm only going to focus on the MinION software, which is what you're viewing right now. In fact, if I go back to the Start menu, this is the software installed, set up, at least at the current version. And you're going to have to take my word for it that I have off camera, and we'll see a little bit later, a MinION flow cell that I'm going to put into the device. This is my first time using this M1KD, and if it's your first time using a device, you'll follow the instructions of the hardware check. There's really not that much to it, so I'll leave you to do that. I'm going to go back over here to the sequencing overview because at the moment I have the serial number essentially of my device, but you'll notice it says there's no flow cell, which is true. So I'm removing a flow cell from its packaging, and you'll see me later insert a flow cell into a MinION device, this very flow cell, this very device. But for right now, that's happening off camera. So I am inserting it, and in a moment it should recognize device, and I believe it will also, yes, recognize the flow cell, which has its own serial number, FBB18251. And it already, since I've done a hardware check, it says that the hardware check is passed. So what I really want to do is check the flow cell. At the moment, I am waiting for some other parts of a protocol to finish. So this is a very good time to check a flow cell. Let me go to the Start menu and click on Flow Cell Check. The ID is already there. The flow cell type is on the back of the flow cell package, and that is correct, Flow Min 114 version. And I'm going to go ahead and press Start. This is also not all that difficult. You insert the flow cell, you check the flow cell. I do want to talk about what is it that we're seeing during a check and what's happening to some extent. So it's just good to document this. I am in the middle, as I mentioned a moment ago, of a library prep. And so I, in some sense, I guess I'm taking a risk by being in the middle of a library prep, and then, as it actually happens in my case, testing my one and only flow cell. If this was a class, or if this is something extremely important, time sensitive, can't mess it up, one, I probably want to have more than one flow cell, but two, I might want to run this check a little bit earlier, but it's not too late at all. If you noticed a moment ago on the screen, there was a little bit of flashing saying that it reached 37 degrees. If I click in here, I am going to get access to some additional view of what is going on. The way that this works, and just to go back to the sequencing overview and then going and clicking on this diagram of the flow cell, those two large rectangles, each one of them filled with squares, are essentially representing an array of the electronics that are embedded in the flow cell itself. The part I think is properly referred to as the ASIC controller. But each one of those little squares, the smaller ones embedded in the rectangles, are essentially representing potential regions, sectors, maybe there's a more specific name, where there could be nanopores and where there could be electrical readings obtained as we go through the actual process of really sequencing. During this point, when it says that, well, this is a little bit misleading, the run state says sequencing, but during this point, when we're checking a flow cell, essentially what is going on is, well, if I move past it, this graph was disappeared, showing me the history of the temperature, which you expect that is kept constant. A moment ago, you would have seen, and maybe we'll see again, the voltage. But essentially, this is one big test of the electronics. I can make this full screen. I can make this less than full screen. You'll see stuff like this a little bit later. But it's going to go through periods of, as it is just now, becoming really active and flashing. Yeah, that's right. They call these little sectors channels. But right now it is testing the electronics, testing the voltage, and if I go back to the overview, it's still making progress on the check. This is going to take several minutes. This is the first time I'm using this device, so I don't know if it's quicker or slower than previous versions, but in my experience, this can easily take somewhere between 5 and 10 minutes for the flow cell check to happen. I also say that it's important for you to check the flow cell before you do any loading, because if the flow cell is damaged, that will impact whether you want to load a library or not, or contact Nanopore if the flow cell comes back as needing a warranty. In this menu, which I haven't pulled up before, called Experiment, it's also another way of visualizing this information. In fact, this is where it's going to tell me, one of the places it's going to tell me how many active pores, nanopores there are once the check is complete. We're still probably a minute or so at least away from that happening. So at this point, I think I've said everything that I want to say about the flow cell check. I'll quickly make a comment once the check is complete, so that we can just comment on what we find. Hopefully that there are lots of nanopores, and this checking process will happen once again when we actually go to sequence. At that time, there will be a check of the flow cell before the sequencing run is started. So it's also, as you'll probably see, normal to lose some pores. Hopefully not too many, but we'll get there when we get there. So I'll make a comment once this flow cell check is complete. Okay, and the flow cell check is complete, and that is a great number, 1,422. I think the minimum is somewhere around 800, so that's looking good. When we look, it tells us it's complete. There is some record of the test that doesn't really tell you very much, but the main thing is that the flow cell check is complete. This little green check says that we're definitely within warranty, and 1,400 or so pores is really a lot. The more pores that are successfully able to capture information, the higher throughput we have a chance of obtaining for our sequencing experiment. So that's it. We'll come back when it's time to actually start sequencing. Okay, so we are ready to go ahead and try to sequence the bacterial isolates. If I go to the overview, I've got my flow cell. It says 1422, but it is going to run another flow cell check because that was from last time. So we're going to say start sequencing, and we're going to call this the 2024. This was the sequence of genome. 2024 sequence of genome isolates. For 19 and 20. I'm going to put I4. Otherwise it sounds like a date. Isolate 4, isolate 19, and isolate 20. And I'll write it down there. 2024 SAG I4 I18 by 20. We got to tell it what kit. This is DNA PCR free multiplexing, and that turns it down because it is in fact the native barcoding sequencing kit 24. All right, and base calling. So all of this different things, we've got to make some choices. So we are going to leave it on fast base calling. We can always read base call later. We have barcoding, so I'm going to leave everything as it is. I want modified bases off for now, duplex off for now. Barcoding, I don't want to trim it because I want to be able later on I can do that myself. I don't know if there are any advanced, yeah, I'm not going to do anything there. It's going to run for 20, I'm not going to run it for 72 hours, but I'll leave it there. let me just change the output. Oh, that's gonna be fine. I'll put it on my desktop where everything important goes. We are going to get the FASTQ files. We are going to keep the pod5 file so that we can redo this. I don't necessarily need to split the pod5 files by barcode, but I'm going to go ahead and do that because it might make some of the other analyses easier downstream. I'm going to save this, and I am going to start. So it's going to take a few minutes to do a flow cell check. I'm not really going to have anything to say until that is done. I can click on sequencing now. It's going to give me some outputs, but essentially if I go back to sequencing overview and click here, there's nothing to see just yet. In a few moments, it tells me it's reached the temperature. In a few moments, we'll be able to see it, even though it's only still just scanning. And it'll take another 5 to 10 minutes in my experience to actually go ahead and really start sequencing. And usually within the first few minutes, you have a sense of, does this look like a good run? And from experience, you start to know what that looks like. So we'll come back. I'll have something to say in a few minutes. Oh, well, since we've gone to active, even though that's not true, I believe I can click on it. And I can start to look at what the different menus will be. So right now, this is sort of the home screen for the detailed look at the experiment. I've got the name, sample ID, total runtime is not very much right now, temperature, read count, which is nothing. On the next screen, it's performing that pore scan. So it's checking the number of pores to see where it's getting good electrical signal and accurate data. Ultimately, we're going to see some output of passed and failed reads. That'll be very important to look at, especially in the first few minutes to tell if things are going well. Then we'll start to get relink histograms, Q-score histograms, barcode hits. So we'll go over that. But for right now, I'll leave it here. This is the prettiest screen, and we'll watch what it looks like when it actually starts sequencing. So we'll check back in a few moments. Okay, so we've got sequencing now. And we've definitely got, I like to think of it, or I think it looks like a bit of a disco. And to some extent, there's some information there. But let me go back to the home screen because we say run state is sequencing. So back here, what do we have? It is been about not quite 10 minutes. And so far, we have obtained around 6,000 reads. And the estimated amount of data is around 21 million bases. So that could be good. Imagine that an E. coli genome is around, almost 4.5 million. And then to actually do a good assembly, oftentimes you need anywhere from 30 times to 100 times coverage. So in 10 minutes, we're on our way to potentially generating a lot of data. One of the things we're also already seeing is the N50. So what is the average length? And this is what I would expect for this protocol, somewhere around in the 20s of kilobases. So let's go back. The pore, there's some detailed things here describing exactly what is being captured at the moment. I'm not going to go too deep into that. One can take a look. But this one, cumulative output, this is really important. So there is a little bit of quality control at this stage. And what you really want to see is that the passed reads vastly outnumber the amount of fails. If you move your mouse, you will get some indication of what the proportion is. So around almost 7,000 passed and about 600 failed. And that tells you generally that we're doing a good job. When that is not the case, when the failed reads outnumber or equal pass, it's oftentimes that you've used the wrong settings or there's some kind of mismatch with what is being, what settings you're using. Something is really wrong and you usually have to stop and figure out what it is. As we continue going on, We get a histogram. There's always some, peak of shorter things that just kind of got chopped up. But by and large, as we look around, we're starting to see a little bump here. And this is good. I think this would be something to assemble. We could hide the outliers and could see some things like there's 100 kilobase read. That's really big. But by and large, it's only been 10 minutes, so we'll keep going. The Q-score, this is also around what we were expecting. For simplex reads, single reads, that we're in the 10s, 12s, 14s. That's really nice. Ultimately, with error correction, we will get something that's really good. And this is the other thing. This is really good. It's also quite informative and interesting. So this is the number of barcodes that we know that sample, if I've done everything right, sample 4 is 1. That was the one that I thought was so viscous and was like having a large trouble. And then later on, barcode 20 became really, really viscous. So clearly, the amount of reads are not evenly distributed across the 3 different samples, 3 different isolates. We can live with that. I mean, we would have liked them to be equal, but clearly, even at this stage, barcode 1 is probably going to do the best. But as long as we keep this running and it keeps doing well, we should get some good data that we can look at all of these. We haven't seen any barcodes that were not used, and I'll just tell you up front, it's not unusual that you'll find a handful of barcodes hits, which are really just sequencing errors, but several hundred barcodes and the ones that we expect, in this case several thousand, that all makes sense. So there's really nothing to do except let it run. So it's been all of 12 minutes. We've generated more sequence than in some decades, certainly some years, and then we'll let that run and come back and take a look at this sometime from now. But this looks like it's on its way to being a good run. Okay, so here we are. We are about 15 hours, it looks like, into a run. That just happens to be when I'm taking a look. And there are a number of things, but first, I'll come back to the screen. Let me just go to the overview. So typically, while you are sequencing, you're going to get something of almost like a little health bar. What you'll see is that at this point, number of hours in, it's actually going pretty well, I would argue, where about 22% of the nanopores are unavailable. In other words, they become clogged, the electrical readings are no longer reliable or usable. And this health bar, unfortunately, will continue to go down. There are things that you can do like washing, and you can add some more library and flush and things like that. But eventually, the flow cell is a finite resource. But seeing it at 62%, that's good. That means most of it's sequencing. There's some pores that are left in reserve, perhaps available. This is pretty good. But you'll notice recursively crashing. What you would not want to see is that an hour or whatever short amount of time into the process, you're almost nothing is happening. That may mean that there is some problem with the library preparation. I didn't pay attention to it or give you too much insight before, but on the system messages, there are often regular updates of what's going on. So you could see these messages happening occasionally, how many pores are available for sequencing. Again, the number of pores will go down. You also get some warnings here. For example, things like temperature or disk space and things like that. So occasionally you want to pay attention to that. But let's go back and click and get some detail on what we've had about 15 hours in. I don't know that this will run for 3 days, but I'll let it run, and we'll eventually, it will taper off, and if possible, we'll see what that looks like then. So for right now, it's the total runtime is 15 hours. The read count is 809,000. Be nice to see that get up to 1 million reads. It's awesome. We also have the estimated number of bases and base called bases. So the estimate is just what it says, estimate about how many bases there are and what the size are, but the base calling, or the process of turning the electrical signal, called a squiggle, which comes and is archived in your POD5 files, into a base call, AC, T, or G, that takes GPU usually, or you can do CPU much slower, but we're working on a MacBook that does have a GPU core, so we're using fast base calling, but essentially we have here an estimate of about 7 or so billion bases, and approximately the size of the fragments around 30 something, which is what I expect. At least these are numbers that I've seen similar to the bacterial protocol, the no-miss protocol on the Nanopore website. So I can't say I'm super unhappy with that. That looks all right. As we move along, we're seeing what we want to see, which is a pretty bright green field of sequencing going on. So that is good. We could always enlarge that, or we could come back and look at different figures. Some may or may not be seen. So you have the option here to look at metrics for different things. The ones that are by default are usually pretty standard, so I'm not going to dig too deeply into that. I'm just going to leave it as it is. All right, so cancel. All right, so let's take a look at some of the other things. Here is our cumulative output, and it is what we want to see, that the number of failed reads continues to be a small subset. You can also do by bases, but gives you pretty similar output. If we go to the histogram, this is important. So here we're looking at lengths, and we're seeing the estimated read lengths, and we have a nice big, there's always some garbage that kind of gets sequenced, but we got a pretty nice distribution with a few longer reads, but everything is sort of in that distribution. Definitely there are ways to get longer reads. I was optimizing for the long reads, longest possible reads, but there are ways. This is showing lengths and estimated. You could also do counts. So if you wanted to know of your reads, where do they fall by counts, there's a histogram essentially. If I don't hide the outliers, you can also get to see a couple of things that might have made it through. So down there is at least one read that's 1.68 million bases, so it's always fun to look at those. I think they're called whales. Let's continue. Okay, here's our Q-score, hovering around 13, 14. That's kind of what I would expect. I haven't done it here, but you do have the ability sometimes to change your zoom. I don't know that it's so impressive here. More on the other one where you can zoom in and sort of see the distribution in a little bit more detail. And then the final one is interesting. Okay, so if you remember when we started, we said there might occasionally be some calls like a barcode 13 and 10. Those are not likely to be real, more sequencing errors. We see here clearly the amount differs in the number of reads, 47,000 reads for barcode 1 versus 58,000 reads for barcode 3, sample isolate 3. Interestingly, if you turn this to bases, it's different. So maybe that's about the sharing. So barcode 2 has 3.5 gigabases, 2 gigabases, and 1.1 gigabase. So interesting when you look at it that way in terms of the total amount of DNA. You can look at it in those different ways. So that's really what I wanted to show you. If I were to close this, the menu shows again is a different way of viewing the results and the information. This is also where we might come to stop or pause. I'm not going to go into all the details, but when it's time to stop it, this will be the place to come and do that. There is going to be a run report. Right now, I guess it would be an intermediate one, but at the end, we'll certainly get a PDF, and we'll review that when the time comes. But for right now, I'm just enjoying that we've got some data coming off, and likely a good chance at doing some good assemblies when the time comes. So I'll leave it at that. Okay, so we are at the end of this experiment, you could see that it ran for 3 days, and we generated 2.28 million reads at approximately 19 billion bases. The flow cells basically died and removed from the device. If we click on this, we can go just into a little bit of depth. We sort of see, it's in sort of tangential process, or as it reaches a plateau, it just sort of levels out, and there's not really much more sequence beyond that point, and the experiment ends there. Again, there are things that you could do during the experiment. You could load more library, you can wash it. Those will give you a bit of a boost. It wasn't necessary, or even in my case possible, since I had to let this run over the weekend. But there are things that you could do to optimize. If we look at the final look here at the output, our peak is still what we've seen sort of early on. You can always zoom in to see a little bit more of the spread. You could hide the outliers and see, maybe we'll try to fish out that 1.93 million base read. If we go and look at the Q-scores, it's sort of what we would expect. And if we look at the barcode sequences, 9 was 154,000 independent reads, versus 1.3 million. We look at the bases, it's a little bit different. So there are fewer, let's see, for barcode 2 as an example, if we go back to reads, there were 524,000 reads, but many more bases. So for whatever reason, these were longer pieces of DNA would be my guess there. You can also see that for other things that we don't really have, let me go by reads, because that's better. There was one read that registered as barcode 4, 16 that registered as 10. These are all not contaminations, but these are miscalls, which is actually pretty good, because out of millions of reads, it accidentally miscalled something as a 20, which wouldn't have been. So the one thing I didn't show, but I think is somewhat intuitive, is that at any time during this, you could press stop or pause to stop the experiment and pause the experiment. It's fairly intuitive. The only thing I'm not showing you here that would come up during that point is that when you stop the experiment, oftentimes the base calling is still happening as the sequencing is going on, and you'll get a choice. Usually you may want to stop the experiment but let the base call continuing. As long as you're using the fast base calling, we happen to be on a Mac, which has a pretty good system, including a GPU. It sequences at the fast level pretty accurately. When I first started this with lower computational powers, there's times when it took days to finish the base calling, so you'll want to be careful about that. If we go to the run report, it will already generate a run report. I don't need to do this, but I guess I am, because I just wanted to be able to show it to you without digging around for it. You will get an HTML, and I don't know if you also get a PDF, but you'll certainly get this HTML report that gives you an overall explanation of everything that you've done, all of your settings, your software, which is really important to know. For example, what version of Dorado, the base caller that was used. But it basically gives you a really nice summary. You can toggle a little bit because this is an HTML file. It's giving you all of the things that you saw or could see in those reports. There's some things that I did not show you, so you could dig in. There's some other reports. You could open up the settings and see Q-score over time. You could see clearly that the health of the pores, the amount of available ones, definitely decreases in that fashion. So those things are all there. These are really great metadata that, of course, you'll want to keep when you're done. So now you're ready to work with the data. I think, let's see if it tells me here. Well, it will tell me that this is on my desktop. Okay, I did that purposely just to make it a little bit easier. So let me open up the folder. Let's do it this way. Just in case, this is your first run or you want to be familiar, you get a folder. This one has to have something associated with the date, but I chose to save it on the desktop. I could tell, for example, it does have some information like what flow cell is there. The key things here are all of these files. So there are a number of other reports, including this one. There's a copy of that report. There's some other things which I'm not going to go into, and mostly are useful for really in-depth troubleshooting with the technician. The key things for you are you get a series, in our case, because we chose to get FASTQ files. Those are the base called files, the A's, C's, T's, and G's are there, but also the POD5 files, and these are the proprietary format. They have extra information. There's information there about base modifications. There's other information that if you wanted to, if there was a new version of Dorado base caller, we could recall the data and perhaps get something that looks even more accurate. So this is really the raw data, and if you really think you're going to come back to do base calling again, if the experiment's really important, I highly suggest, as long as you have the memory to save those. But immediately, and or if you're not going to come back to the experiment, you're just running it once, see what you get, you may only want the FASTQ files, and in the case of this, it's the pass, that we're most interested in. Fail are FASTQ files that did not pass quality control. But the pass folder is here. You get a folder of unclassified, so there are a very small amount of sequences, even though it does look like there are many folders here, where it could not classify the barcode. So those are here. There are a small number of files in these that, for example, this is barcode 10. These will all be a small number of files. There's nothing really in there, but and the ones that we are interested in, which is 1, 2, and 3, there will really be a lot of data. So these are all of our files. Each one of these, I believe each one of these zipped FASTQ files contains somewhere around 4,000 reads, if memory serves me well. But these are all the basis for analysis. So that's it. As far as the generating of the data, the next part of bioinformatics would be, in our case, doing some additional quality control on the sequence, remove barcodes, adapters, remove that control spike in of the DCS, that phagment, I think it's phage DNA that we added, but then going ahead and doing an assembly. So that's it.